So happy to be here to talk about the basics of forensic investigative genetic genealogy. Just a little bit about myself and how I got into genealogy. A few years ago, almost 10 years ago now, my great aunt posted this newspaper clipping to Facebook, and it's about my family who had served during World War II. This was an article that was written during World War II. And in the article, it also mentioned my great-great-grandfather who had served during World War I. And I wasn't aware that I had family that had served, and I just got really curious about my family history. I fell down the genealogy rabbit hole, as I'm sure many people watching are familiar with that, when you just get so interested and you want to learn everything you can about your family history. As part of that, I did a DNA test, wanted to learn how DNA could help me with my genealogy research. And I just find DNA super fascinating, wanted to learn everything I could about it. And on the way, I became a search angel. So I'm helping to reunite adoptees with their biological family. And since that work is very similar to FIGG work, I did, as mentioned, work with Rae-Venter for 2 years, working on forensic cases in genealogy. And that's when I did the course at the University of New Haven and really enjoyed that. And so when the opportunity opened up, I was able to now teach there, which I also really enjoy. So usually when we talk about FIGG, we're talking about, everyone sort of sees the beginning as the arrest of Joseph DeAngelo, who was also known as the Golden State Killer. He was a serial killer. He was active between 1974 and 1986 in California. He committed 13 homicides and over 50 rapes, and he burglarized 120 homes. And this really was, created a really big splash in the genealogy world and also in the law enforcement world, because now we have a new technique that we can use to solve cold cases. But one thing that we have to know is that that actually wasn't the very first time that FIGG was used. 2 weeks before that, the DNA Dope Project had announced the identification of someone known as the Buckskin Girl. And I guess unidentified human remains are not as exciting as serial killers. And so it really, I don't think, caused the same kind of splash that the announcement of the Golden State Killer arrest did. But what I think I want people to understand about this is that there's no one person or one thing that was the beginning of this. There were a lot of genealogists kind of all thinking the same thing, working on different cases. And the Golden State Killer happened to be the one that got announced first, that made the biggest splash. And that's what people tend to think of. But we can even go even further back in time. And we can talk about Lisa. The Lisa Project was the precursor to the arrest of the Golden State Killer. In 1986, a man was arrested in California, and he had abandoned this little girl named Lisa. And then in 2002, he was arrested again, this time for murder. He was using a pseudonym, but using DNA, they were able to see that this was the same person. And that was when they were able to discover that this was not Lisa's biological father. So the question was, who is Lisa? Where did she come from? It really wasn't until 2013, when the public DNA databases were a lot more popular, there were a lot more people in them, that the detectives decided they were going to try to figure out who Lisa was. They went to an organization called DNA Adoption, and there was a genealogist working there named Rae-Venter. And so they were able to work, they had a whole team working on it. They discovered that Lisa's mother was Denise Beaudoin, and she had been missing. She went to go visit her family in New Hampshire, and nobody saw her after that. Around the same time that Lisa was abandoned, New Hampshire detectives found the bodies of a woman and 3 children in barrels in Bear Brook State Park. And now that woman in the barrel, that was not Lisa's mother, was not Denise Beaudoin, but they did discover that the man was the biological father of one of the children. And they were like, who is this man? And they were able to use genetic genealogy to determine that his real identity was Terry Rasmussen. And the detective thought, if we can identify people just using their DNA, then why can't we take DNA from the crime scene and use that to identify people? So they took DNA from the scene of the crime of the Golden State Killer, and they, the rest, as they say, is history. So since that time, we have had, I think, almost 1,500 cases that we know about. These are cases that have been publicized. There is somebody keeping track every time an announcement is made in the news of a case that gets solved, whether it is a suspect case or unidentified human remains. And so we can see that this is a very valuable technique to identify people, to identify suspects, but also the unidentified human remains, to give them their names back. And it's something that is becoming increasingly popular, and as its popularity grows, increasingly more accessible to law enforcement. So before we go to talk about, how does this all work, we have to start by talking about DNA. So there are different types of DNA. We have our autosomal DNA that we inherit from all of our ancestors. We have our sex chromosomal DNA, X and Y DNA, our X DNA, which is inherited in specific patterns from specific ancestors, and our Y DNA, which traces the paternal line going all the way back. We also have mitochondrial DNA, which we inherit from our mother, and so that traces our maternal line going all the way back. And even when it comes to FIGG, we're talking mostly about autosomal DNA. We do use Y DNA, and even going back, we have used Y DNA, if you want to consider that FIGG, where they use Y DNA and not autosomal DNA. Less so mitochondrial DNA, but in some cases, mitochondrial DNA doesn't degrade as quickly, so it is very useful. For example, the case of King Richard, who was buried, they found him under a car park in London, and they were able to identify him using his mitochondrial DNA. So we can use all of these DNA to identify people, but when we're talking about FIGG, more often than not, we're talking about autosomal DNA. And even when it comes to autosomal DNA, there are two ways that autosomal DNA is analyzed, and they really almost creates two distinct types of DNA because you can't compare them to each other. They're very different, apples and oranges. So traditional forensic DNA analysis involves what we call STRs, short tandem repeats. And so as you can see here, we're looking at all these nucleotides, and there's these little repeats here. And we're distinguishing one person from another person by saying this person has 7 repeats and this person has 9 repeats. Now, it's possible for 2 people to have the same number of repeats at a particular location, but when we start looking at it at, say, 20 different locations, the odds of 2 people sharing exactly the same number of repeats at all 20 locations is something like 1 in a billion. So it is very, very identifying, and this is what we have used traditionally in DNA forensic analysis. Now, when we're talking about DNA testing, not just even FIGG, but any kind of DNA test that you might take for yourself, spit in a tube or swab your cheek and send it off to a company, well, we're talking about what we call SNPs, single nucleotide polymorphisms. And what we're looking at here is not repeats. We're looking at every single letter, and we're looking for segments where all of these letters are the same. And that means that when we have these segments, these 2 people are related to each other. And obviously, the longer the segments are, the more segments you have, the more DNA you share, the more closely you are related to someone. So the main differences between STRs and SNPs, if we're talking about STRs, we're talking about 20 places on the genome, depending on the government and how they decide to do it. We're talking about government databases. So in Canada, we have our national DNA database. In the United States, you have what's known as CODIS. You also have state databases. And we really think of STRs as being identifying the same way a fingerprint's identifying. It lets you know who someone is, can distinguish someone from someone else, but it's really not telling you that much else about that person. Whereas when we're talking about SNPs, we're looking at about 300,000 to 600,000 places in our genome. We're talking about public DNA databases, although if we're talking about FIGG, we're talking about GEDmatch and Family Tree DNA, because those are the only 2 databases that allow for law enforcement use. And we're talking about our biographical core, really like everything that makes us who we are is in our SNPs, our appearance, everything medically relevant about us, and our relatedness to other people. All of that is in that DNA. Now we measure SNPs. We use a term called centimorgans, a unit of measurement. Each person has approximately 3,400 6,800 centimorgans of DNA, 3,400 that they receive from each parent. And so coming down from there, I would say my full sibling and I, we share about 2,500 centimorgans of DNA. A first cousin and I would share around 900 centimorgans of DNA. And you don't have to memorize all of these. We have a lot of tools, particularly one that I use all the time, the DNA Painter Shared Centimorgan Project. And all you have to do is take the amount of centimorgans that the company gives you, pop it into the chart, and then it'll tell you very likely what the relationships are. Now, the closer you are related to somebody, the easier it is to tell what the relationship is. If somebody shares 2,500 centimorgans of DNA with me, they're my full sibling. That's really the only relationship that's possible at that level of DNA. But if I said somebody shares 10 centimorgans of DNA with me, well, they could be a third cousin, they could be a fourth cousin, they could be all the way out to an eighth cousin, maybe even beyond a genealogical time frame. I may not be able to identify them. And so what is important to know from that is that when we are doing FIGG, we want to have DNA matches who are closer related to us. It really makes our job a lot easier in identifying the potential relationships between people. Now, how do we do this work? So we make a SNP profile, a DNA profile, we get a list of DNA matches, and what we're going to do is we are going to build back our DNA matches until we find what we call a most recent common ancestor, an MRCA. And then once we have found that most recent common ancestor, then we got to start building down and finding the person who that unknown DNA belongs to. And so you can imagine that, I mean, everyone who has experience with building just their own family tree knows how time consuming it can be to build a family tree. So now we're not doing that just for one person, we're doing that for multiple people, and not just building all the way back, but also all the way down. So you can see how resource intensive FIGG is. So how do we find that most recent common ancestor? Well, the first thing we want to do is harvest the potential of the genetic network. Now that sounds like a really complicated term, but really all it means is I have these green DNA matches, they match each other, and they match our unknown DNA. And so this makes a genetic network. So we're going to try to find their most recent common ancestor. So who are their parents? Who are their grandparents? Who are their great-grandparents? And sometimes we have to go even further beyond that. But in this case, these two DNA matches share great-grandparents, and that is their most recent common ancestor. Now, what we would do is, depending on how close these DNA matches are to each other, we would either have to build back a little bit on this line before we started building down. But let's say they share an amount of DNA that is consistent with them being second cousins, that this unknown DNA match would also be descended from these great-grandparents. We could just start building down from these great-grandparents and try to figure out which one of those descendants our unknown DNA belongs to. Now, even better than one genetic network is two genetic networks. If we can harvest the potential of multiple genetic networks, that's going to make our life a lot easier. And so we have this green genetic network here and this pink genetic network here. The green genetic network shares DNA with each other and our unknown DNA. And the pink genetic network shares DNA with each other and the unknown DNA. But the green people and the pink people, they don't share DNA. They're not related to each other at all. And so what do we do with a genetic network? We build back until we find that most recent common ancestor, and we do it for all the genetic networks we can identify. And then we build down again. And sometimes when we are building down, we can find, for example, that somebody from the pink family married somebody from the green family. So they're not related to each other at all, but now they're connected to each other through what we call a union couple. And that is so good to find, because once we have identified this union couple, all we have to do is build down from them. We don't need to build down from these MRCAs back here. We can be building down from this union couple down here. And the more union couples we have to build down from, the more it really narrows down who it is that we're looking for makes our life a lot easier doing this work. So what do we call this tool? I've been calling it FIGG, and I sort of want to make it clear as to why I use that term. There are a lot of different names that I have heard for it. If you're reading government documents, you're likely to see Forensic Genetic Genealogical DNA Analysis and Searching, which is a very big term, mouthful of a term. If you're reading a news article, you may hear something like genealogical DNA or advanced DNA techniques. And I don't find that those are particularly helpful. They don't really specify what kind of advanced DNA technique they use. So I'm not really liking those terms. And I wish newspaper people would watch, webinars like these and educate themselves on the topic so they understand how it works. We don't want to call it forensic genealogy. Forensic genealogy is a field that has been around for a very long time. It involves cases that do go to court, and so that's generally why people sort of associate it with that way, that forensic genealogy, but it doesn't necessarily have to involve DNA. So you can consider FIGG to be sort of under that umbrella of forensic genealogy, but it is not the same as forensic genealogy. Forensic genealogy refers to things, for example, like probate cases or military repatriation, stuff that doesn't necessarily have to involve DNA. And if we want to be more specific about what we're referring to, we can't just call it forensic genealogy. And familial DNA searching is another term that we cannot use because that is referring to a completely different technique. It's similar, and that's why I guess why people conflate the two terms, and we'll talk about them in just a minute. But suffice to say that they're different enough, and we don't want to be conflating them. We could just call it genetic genealogy. Genetic genealogy is, again, it's too broad a term. I use genetic genealogy for my own personal genealogy research. It's not specific to the type of work that we're doing with FIGG. Paul Holes, who was the detective on the Golden State Killer case, coined the term investigative genetic genealogy. And that's a term that has stuck with a lot of people who still use that term. But the people in the forensic community preferred forensic genetic genealogy because we really want to understand that even though we're not in a lab and we're not working with biological samples, the DNA profile that is created from that biological sample is forensic evidence. And so everything that we are doing with that profile, all the tests we're running, all those DNA matches, all those trees that we're building, that's forensic evidence that has to stand up in court. And so we don't want to forget that forensic aspect, which is why I prefer the term forensic genetic genealogy. My boss, Dr. Claire Glynn, who is the director of the program at the University of New Haven, the graduate certificate in Forensic Investigative Genetic Genealogy, wrote a paper that she said, you know what, let's split the difference. Let's call it Forensic Investigative Genetic Genealogy to appease both the IgG people and the FGG people. And why not? It has a really great acronym, FIGG. I think that makes it very easy to talk about. We don't have to say the whole thing. We can just call it FIGG. And so those two last ones are the preferred terms for myself to talk about what this is. And if you are interested in reading more about that, there is a link in your handout to this article that I wrote for the issue report, What's in a Name, that talks about, that basically kind of what I went over, why the particular names matter. So I said that I would talk about the difference between familial searching and FIGG. So familial searching is using STRs and FIGG is using SNPs. So already right there, you can see, we already know that STRs and SNPs, apples and oranges, two completely different things. When we're using familial searching, we're talking about government databases where allowed. So the CODIS or the national database in the United States, you can't do familial searching there, but in certain states where it's allowed, those state DNA databases, that's where the familial searching will take place. Whereas with FIGG, we're talking about public databases, and we're only talking about GEDmatch and family tree DNA because those are the only databases that are authorized for law enforcement use. With familial searching, you have to have authorized personnel, the people who are allowed to go into those databases and perform those searches. Whereas with FIGG, pretty much anybody could hang up their shingle and say, yeah, I'm a FIGG practitioner. When we're talking about familial searching, it's only possible to find parents, children, siblings of a person we're looking for. Whereas when we're talking about FIGG, we can have that immediate family first cousins, second cousins, third cousins. I think people have even solved cases using fourth cousins. So it really is much more far-reaching. And we want to make sure now that we understand the difference between these two things. They're both involving families and DNA, but they're really very, very different techniques. So let's talk about the typical FIGG case workflow. How does it work? We start with picking a case, and we want to make sure that we're looking at violent crimes or unidentified human remains. We want to make sure that the cases that we are picking are going to match up with the terms and services of the websites that we are allowed to use. And then we want to make sure that we've done an STR profile. If I can just upload the STR profile to a database and get a hit there right away, why would I go to the trouble of FIGG? You can see how resource intensive it is, how time consuming it is. We want to do the STR profile first. We want to make sure we have DNA. You know, we use up DNA every time we analyze it and make a profile out of it. Do we have DNA remaining from the case? Do we need to go back to the evidence and reprocess it so that we can get more DNA to work with? And when we're looking at our DNA, we want to know, what is the quantity? What is the quality of that DNA? We want to make sure we have enough to be able to get a good DNA profile, and the quality of that DNA is going to inform us about what SNP technology we're going to use to analyze that DNA. So for example, microarray is a very standard form of SNP analysis. It's pretty much what all the companies use. If you spit in a tube or swab, send it off to them, they will do a microarray and analyze your DNA. And if you have DNA that's in a good quantity and a good quality, a microarray is probably what you would want to do. But let's say you have a body that was found in the woods and it's very badly degraded, the DNA, and it maybe even has bacterial contamination. Then you might want to do something like whole genome sequencing. And there is also what we call a targeted SNP kit. This is something that Verogen has come out with. They call it Kintelligence. And instead of that 300 to 600,000 SNPs, they've narrowed this amount of SNPs down to about 12,000. Only the relationship SNPs, none of that medical appearance related stuff. So very targeted. And the nice thing about the targeted SNP kits is that you can buy all of the stuff and bring it in-house, make it part of your in-house workflow, which may work very well for some law enforcement agencies who want to be doing this work in-house. And no matter which method you choose, we're going to have to do bioinformatics work on it, basically kind of massage the data and make sure that we can get a usable DNA profile, because we have to upload that. It has to be accepted, the upload, and fit into the algorithms of FTDNA and GEDmatch Pro. Now, we can't just upload our DNA the same way I would upload my own DNA or my family's DNA if they want to do a DNA test for me. They have to fill in a lot of forms. GEDmatch Pro is the law enforcement portal, and you have to put your law enforcement cases through that law enforcement portal. And again, the cases have to match those terms and services of those websites. Now, once we get all of that work done, then the genealogist comes in and we start analyzing all the DNA profile using genetic genealogy tools. Basically, that's the entire course that I teach, 7 weeks of learning all of the different tools. What do they do? How can we find those genetic networks? And then we move into genealogy work. The genealogy, we want to collect the records, we want to build the trees, and we want to make sure that we're doing a good job with our genealogy research. I'm sure everybody knows somebody who's out there just clicking on those shaky leaves and adding everybody and anything to their trees, and that's not good enough when we're trying to identify a suspect or identify unidentified human remains. We really want to make sure that when we're done, that we have identified the correct people and that our work is reliable. So if we are able to come to an identity candidate, we want to have somebody look over our work, make sure that we did a really good job, that our conclusion is very solid, so that when we hand it over to law enforcement, they're going to be able to find the right person, and they're not going to put effort into identifying someone that isn't the correct person, because they're going to collect reference DNA from the person we've identified. Now, sometimes they will get a warrant and they will go to that person and say, please give us your DNA. Sometimes they will do it surreptitiously. They'll follow them around until, they drink from a cup and put it down, or wipe their face with a napkin, collect that, and they bring it back to their lab and then analyze it. And we're going back to that STR profile. They are comparing it to the original crime scene DNA using STRs. We know that STRs are well accepted in court. It's very standard. And so it just makes sense to use that in court to say, we know that this person's DNA was found at the crime scene because their STR profile matches the STR profile of the crime scene DNA. SNPs are starting to be used in court and have been very accepted in court, although it is not as common as STR. One day, perhaps, maybe we'll just do everything with SNPs. I don't know. That remains to be seen. But currently, we always go back to that original STR profile to get our confirmation. Now, I'm hoping that as people are listening, they're seeing all of the ethical issues that can arise from doing this work. There's a lot of different things going on here that we want to make sure that we're doing this work ethically. A lot of people, when they give presentations on FIGG, they're like, oh, it's the Wild West and everybody's just kind of doing everything. It's completely unregulated. We just do whatever and hope for the best. And I kind of feel like that doesn't really capture the danger of that situation. I really prefer the analogy of building the plane while we are flying it, because that's, you sort of get that idea, like we're up in the air and our plane isn't really fully created yet. And we're hoping that we're going to be safe while we're doing this. We really have to think about a lot of different ethical considerations. I'm just saying just the tip of the iceberg, things that I can think of. I gave an entire lecture that was just about ethical issues in FIGG. So I just got to highlight a few of them. Things like informed consent. If I upload my DNA to this website and then I check off, yes, I'd like, law enforcement to be able to access my DNA profile for comparison against their DNA profile that they uploaded. It's a lot like, the terms and services, where we just click, yep, I read the terms and services, but nobody ever reads the terms and services. They just check the box so they can, move on. And so does that really inform consent? Do I really understand what I'm consenting to when I'm giving my DNA or allowing law enforcement to have access to my profile if I happen to be a match to their kit? Security. Like I said, DNA profile is forensic evidence. All of the tools that I use and the results, all of the trees that I'm building and all of that, that's forensic evidence. And if I'm doing that and it's on my computer at home, who else has access to that computer? Is that safe? We want to think about things like impact on minoritized groups, accreditation of genealogists and laboratories. I'm going to talk a bit more about accreditation of genealogists later, but although some of the work we are able to bring in-house in law enforcement, for the most part, we are using private laboratories to get this work done. And so are those laboratories accredited? Are they capable of producing work that's going to stand up in court? And same thing with the genealogists. Is this just an amateur genealogist? Can they be qualified as an expert witness if they need to? And if all of this is brought to court, is that going to pass in court and get to the point where we can have the results that we want in court? Jurisdictional issues. A lot of people like to kind of put their head in the sand and pretend that this doesn't exist. But like, I'm in Canada. Let's say I was on a trip to the United States and something happened to me and they found me and I had no ID on me or anything. They have to identify me. And so I have DNA matches that are in Australia, that are in the UK, and then they're going to find, maybe DNA matches in Canada. And if they narrow in closer, now they're going to have to talk to family members in Canada across the border. And every single one of these jurisdictions has different rules, regulations, privacy laws. That are we respecting all of them as we work this case? Privacy and confidentiality, even within one jurisdiction, so many states now are putting out privacy laws. Are we following those and making sure that our work is compliant with that? Same with confidentiality. If we have to get, for example, a reference tester, we need somebody who we don't think is a suspect, but maybe is closely related to the suspect, and we want to see, is this person actually related? We may ask people, can you do a DNA test? And we just want to see, you know, where this person fits into your family tree. It has happened that, and it was very big news that, you know, they talked to somebody about a case, and now she's on TikTok telling everybody that she did a DNA test and now her grandma got arrested. Do we really want that for our cases? You know, we're going to be compromising cases again that maybe have to go to court, and now they're compromised because somebody is talking about the work that we are doing that they shouldn't be talking about. And even amongst, you know, the labs and the genealogists and things like that, how are we making sure that that confidentiality is being maintained? We want to talk about things like the types of cases are allowed. Violent crimes is a very broad term. Is kidnapping a violent crime? Is stalking a violent crime? You know, we have to kind of draw a line in the sand and say, this is what we are using your DNA for. And we can't just say, oh no, wait, by the way, yeah, now we're going to also use it for this. And we really have to think about, if people, you know, we draw that line in the sand and people cross that line, what are the consequences of that? Because we have to make sure that we're doing this work in an ethical way and holding people accountable so that people are doing this work in an ethical way. The thing about this work is that it relies on DNA matches to get it done. If we had no DNA matches, we could not do this work. And so if I committed a crime and the judge said, put your DNA in that database, I don't have a choice. My DNA is going in that database. But if I'm just a regular person and I'm doing DNA testing, I don't have to put my DNA in GEDmatch or in Family Tree DNA. I may be encouraged to put my DNA in those databases because I feel like helping law enforcement with these cases is something I want to do. But if I feel that law enforcement is not doing this work ethically, responsibly, I'm not going to put my DNA there. In fact, if my DNA is already there, I may take it out. And I'm also going to encourage other people not to put their DNA there and encourage other people to take their DNA away from there. And like I said, we need these DNA matches to make this work. If everybody decides we're not doing it well and they take their DNA out, then we can no longer do this work. And that would be really sad because we've done so much good and helped so many people with the work that we've done here. Conversely, if we do good work and we're showing people, look at all the good things that we're doing with this, people will be more encouraged to put their DNA in these databases. And once our databases grow, we will solve even more cases. I can't tell you how many cases I was working on that just needed one good DNA match to push them over the edge and get that much closer to being solved. Now, we don't need to reinvent the wheel when it comes to ethics and regulations and legislation and all that kind of stuff. There's a lot of great stuff already that exists that we can use to help inform our decisions and help us to be ethical FIGG practitioners. You know, when it comes to doing the genealogy work and the genetic genealogy work, we already have the genealogy standards. We already have genetic genealogy standards. We just need to make sure that we're following them. The United States Department of Justice put out an interim policy that has a lot of stuff that we need to be following. The Scientific Working Group also has guidelines. I also want to mention the National Technology Validation and Implementation Collaborative, the NTVIC. They are made up of people from all over the spectrum, and educators, academics, laboratory people, forensic people, law enforcement people, and they're getting together and they're having these conversations about what do we need to do to make sure that we're doing this work right. And then they're putting out publications talking about all of those things. If you go to the website, everything is there, all the documents that they have put out. Highly recommend reading them. It really is a great organization that I think is going to have a big impact in making sure that this field is done, all the work in this field is done well, responsibly, and ethically. So now we're going to talk about educational opportunities. If you're interested in getting involved in this field, obviously we want to start by educating ourselves and learning how to do this work. And the first thing I want to talk about is that idea of accreditation or certification, because a lot of people think, well, I did this class, and now I have this certificate, and that makes me a certified genealogist. And that is not the case whatsoever. You cannot call yourself a certified genealogist if you have not done a certification. So a certificate is a proof of an education, proof that I took this class, whereas a certification is proof of competency. It is, I am capable of doing this work. You know, I think of it maybe like law school. I can go to law school. I can get my law degree. That's my certificate that says I went to law school, but I'm not able to practice law just with that degree. I have to pass the bar. Passing the bar is a bunch of lawyers evaluating me as a lawyer and saying whether or not I am capable of practicing law. And it's the same thing with genealogy. The reason why you cannot call yourself a certified genealogist just because you got a certificate is that that is a trademark term of the Board for Certification of Genealogists. There is also an organization called ICAT-Gen, and they have accredited genealogists. And these are certifying bodies that have been around for a very long time, and their certifications hold weight. You know, if somebody says, I am a certified genealogist, or I am an accredited genealogist, everyone in the genealogy field knows that this person knows their stuff because they submitted a whole portfolio and somebody evaluated and said, yes, they are a good genealogist. Now, this is only a certifying body for genealogy. If you get that certification from the Board for Certification of Genealogists for genealogy, you can apply then and do a portfolio to get your certified genetic genealogist credential. But even then, neither of these are certifying bodies for FIGG. There is an organization that has recently come out that is giving that kind of a credential, the IGG Accreditation Board. They, like I said, just came out. At this point in time, we don't know whether or not their certification is going to hold the same kind of weight, that the people who are being certified are really going to be, that certification is going to be really meaningful. So we'll see as time passes whether or not that is going to be the case. So a lot of these genealogy and genetic genealogy education things that I'm going to be talking about, there are links in the syllabus to my blog. I wrote 3 different blog posts about education. I did a whole lecture about it at the East Coast Genetic Genealogy Conference. So really I could talk for 45 minutes just about this. But here's just sort of the highlights for you. You could do a formal course like Boston University. The International Institute for Genealogical Studies has certificates, whether it's in a particular geographic location. They also have a certificate in DNA. The Research Like a Pro course takes you sort of from A to Z. How do you do genealogy? They also have a Research Like a Pro with DNA. You could do an institute. So these are week-long, all-day-long courses, SLIG, IGER, GRIP. Really great. They have a wide variety of topics, pretty much anything you ever wanted to know in genealogy and genetic genealogy. There's a course for it. There's a lot of informal learning that you can do. Webinars. Legacy Family Tree has so many webinars that you could watch about anything. The Allen Library here, they have a lot of webinars available on their YouTube channel. Family Search has lots to read and watch. Ancestry has Ancestry Academy. There's tons of books that you can read, blogs, Facebook groups, YouTube, lots and lots and lots of good stuff out there. I also recommend an NGSQ study group. I did those for a few years. The NGSQ is the National Genealogical Society Quarterly. It is a very well-respected journal that when you're reading those articles, you're reading really good genealogy work. Whether it's a case study or a family history, you know that for their article to get accepted, that somebody really did their due diligence. And the NGSQ study groups are just really great because you read an article each month and you have a whole group of people to talk about it with and ask questions and point out things that you maybe wouldn't have thought of. And I really enjoyed that. I'm also a graduate of ProGen. You do have to have a fair bit of genealogy education behind you before you're allowed to join ProGen. But if that is the case, I do recommend it. It was a really great year. I learned so much, and it was a really great way to level up my genealogy skills. And of course, there's lots of conferences. Roots Tech They also have videos online from past conferences, and the National Genealogical Society puts on conferences also, and again, a wide variety of topics related to genealogy and genetic genealogy. Now, in terms of FIGG education, I mean, we're way ahead of where we used to be, but again, it's a very new field, so there really isn't quite as much out there for FIGG. Ramapo College has a program, and again, this is all of these links to these places are in my blog post. University of New Haven, which obviously I'm biased because I teach there, I've done the course myself, but I do think it is a really fantastic program. It's very comprehensive. If you're a genealogist who doesn't know anything about forensics, that's the first course. That's all it is forensic stuff, kind of like CSI stuff. I loved it. It was fun. And then, like I said, genetic genealogy, genealogy, and then the practicum at the end. They give you a GEDmatch kit number, tell us who this person is, and write a report. That's your project for this term. And I think it's a really fabulous program. And there are a lot of conferences. The Ramapo College puts on a conference. The RIG ISHI, the International Symposium for Human Identification, has sometimes workshops or panels about FIGG. And the International Society for Forensic Genetics also does some talks sometimes on topics related to FIGG. I always want to encourage everybody to upload their DNA and opt in. I put a little section on your handout about how you would go about doing that. There's a link to Roberta Estie's blog. It's a fabulous blog post that I always give to everybody because it's how do I get my DNA from this place? And it tells you how to do that. And then how do I get it to that place? It tells you exactly how to do that. There's also a link there to GEDmatch, the GEDmatch education page. And that will tell you how to upload your DNA, how to create a GEDmatch account and upload your DNA there, and opt in. If you want to opt in, if you really believe in what we're doing here and the work that we're trying to accomplish, make sure that you've ticked that box, because a lot of people say, I mean, some people say, oh, I did a test at this company that is not GEDmatch or Family Tree DNA, and I'm helping law enforcement. And I was like, no, you're not at the right place. So your DNA needs to be here. And even if they're here, a lot of people, make sure you double check that. If you have your DNA there, that you have ticked those boxes. I know when GEDmatch came out with its consent for law enforcement, they opted everybody out. And so a lot of people have not yet returned to go back and opt themselves in if that's what they so desire. And that means that actually that some cases that we solved before that happened, if we were having to do those cases right now, we would not be able to solve them because those matches are now gone. So like I said, there's a lot of cases out there that are just one match away from being solved, and that you could be that match. So I do encourage everybody to upload your DNA to those places and opt into law enforcement matching. And with that, if anybody has any questions, I am really happy to answer those questions.
We do have a few questions for you here. So the first one is talking about MyHeritage and how they're not allowing people to upload DNA from other companies any longer. And so this person's wondering if you might be able to speak to that, about how investigative genetic genealogy might have been involved in that decision.
I don't know what's going on at MyHeritage. I don't have any sort of inside scoop into that, but I do suspect that FIGG is the reason for that. And it's very unfortunate, especially for people in the genealogy world who just want to do their genealogy stuff. But it has, I mean, even since the Golden State Killer case, it has come to light that people are using MyHeritage for FIGG when they are not supposed to be. It is against the terms of services. And I think that they have decided that the only way that they can prevent that from happening is just by not allowing uploads, the same way that Ancestry and 23andMe don't allow uploads. And if you can't just, you know, it's very hard to spoof DNA, especially DNA that's, you know, if everybody understood how hard it is to do process DNA, they wouldn't think that people are just mailing in, you know, crime scene DNA samples to Ancestry. It really is. It cuts off, because a DNA profile that you can upload, that's easy to do, you know, but if people aren't going to be following the rules, and that's what they feel is their only chance. I think it's really a shame, but at the same time, I mean, it's better. They understand that it's better for the people who want to maintain their privacy at MyHeritage and don't want to opt into to law enforcement, matching, and they want to keep them private from that, from bad actors who are using that DNA against their consent.
Thank you. Let's see. Somebody mentions hearing about a lot of the great successes in forensic genealogy, and it sounds like they're curious about cases that may not be able to come to a resolution. So, or cases where you just can't find the matches. So,
Yeah.
I think they're curious about that and maybe how you move on from that or move in a different direction.
I mean, the worst case I ever had, I think the highest match was maybe like 10 centimorgans. And I was like, yeah, that case is not getting solved anytime soon. But you never know, and I was working on a search angel case once that was really difficult, had very, very distant matches. And then one day, half sibling popped up, boom, case solved. And so that's why I keep saying, like, all of these cases, they just need that one good match to get them where they're going, and then they'll be solved. So if we can't solve a case, and I think when I was working for Firebird Forensics Group, that's a nonprofit. So we work on cases until we really can't anymore. We put as much time as we can into them. But like companies, for-profit companies will decide before they even take a case whether or not it's worthwhile, or telling the companies or the law enforcement agency, this will cost you a lot of money if you want to solve this case, because we're really going to have to do a lot of work here, and then you're going to have to ask for a lot of reference samples to get us closer to where we're trying to go. But the cases that you cannot solve, especially for a nonprofit group, we kind of just babysit them. They just kind of sit there, and we check every now and again. Did this case get a new match? And if it gets a new match, that's a really exciting day. And it's like, all right, now let's get back to work, and okay, where were we in this? And how does this match fit into what we already know about this case? So, and it is, I think it is kind of almost unfortunate, because it kind of looks like magic what we do. And I don't think people really appreciate, A, the amount of work that goes into it, and B, how unmagical it is when you have probably more cases that you haven't solved than cases that you have solved, because there are really a lot of cases that don't get solved, and we will never hear about them.
Thank you. And speaking of a current case, someone's asking about how important the current Brian Kohlberger case is and its legal decisions to the future of FIGG.
So I'm not following it super closely, and I am sort of keeping it, my eye on it a little bit, especially when it does finally reach its conclusion, because I do agree that it is a really interesting case when it comes to FIGG and how FIGG is done. For one, I think it's a case where most of the cases that we're working on in FIGG are cold cases. They're very old. They happened a long time ago. This is a first instance really where we're using it on an active investigation. And so again, drawing that line of at what point do we decide to use FIGG is interesting. Should we be using it more on active cases and not waiting until it gets to the point of a cold case? That's really interesting. And then I think also the legal aspect of it, it has been tried before. A lot of people have tried to argue, oh, this goes against my constitutional rights, or those kinds of arguments that have not been successful. This one's sort of taking a little bit of a different tact. I think they're also trying to say, oh, you know what, people who are doing this FIGG work aren't doing it very well, and there's been ethical breaches. And you know what, they're right. And I think that's why we need to make sure we're doing this right, because we don't want to give people ammo to say, no, we shouldn't be doing FIGG work, or if you use FIGG work in a court case, now that court case should get thrown out because it's not valid. So yeah, it will be, I think, really interesting to see where this lands.
Thank you. We have a couple of similar questions, so I'm combining these. But do you have any special suggestions or techniques that you use in situations with endogamous populations?
Oh man, yeah, you could do a whole lecture just on endogamy. It's not my specialty. My father's side is endogamous, and work with my mother's side more because that's where more interesting mysteries are, but also because that endogamy is just, it is. I do one whole lecture on that in my course and talking about it. It's really, really hard. And I think what makes it really hard, like when I'm talking about those union couples and how you have, you know, green people who are not related to pink people, but when you have endogamists, that you don't have that. You can't find those genetic networks don't exist because everybody in the network is connected to everybody else. So you can't, those distinct networks, they really don't exist. And so oftentimes when you're dealing with endogamy is you're brute forcing it. You're just building trees as much as you can and getting everybody in there and set. And it really just makes it that much harder. And it really is. It's a lot more work. I know Leah Larkin, she has a blog called The DNA Geek. She offers classes on endogamy. I did do one class with her that I thought was good. And it's helpful for people to get a handle on endogamy and how to work with that. She also was one of the people behind a tool called Banyan DNA, which is a really useful tool when you're building trees with people who kind of may have an ancestor that's shared, you know, pedigree collapse. Basically, you have an ancestor who appears in more than one spot in your family tree, or cousins who are connected in multiple ways. That is a really good tool to use. And there's I believe there's a YouTube showing if you want to learn how to use it, but I think she also offers classes on how to do that. They have a whole Facebook group and meetups and things like that where they talk about, you know, what they're doing with that. So it's just a matter of education and finding the right tools, because a lot of the genetic genealogy tools that we use, for example, the Shared Centimorgan Project, you can't use that when you have endogamy. You know, I have a DNA match. I popped him in there and it said at most this person is a third cousin. It can't be any more distant than that. And when I did the research to figure out how are we actually connected, they're actually my fifth or sixth cousin. But because they're my fifth or sixth cousin in 3 different ways, that DNA got all piled up and it makes them look like they're a third cousin. So having the right tools and just education, looking for educational opportunities that are going to help you learn more about endogamy and how to get through it.
Helpful, thank you. We have just a couple more questions, but I'll go ahead and mention if anybody would like a copy of the chat or if you have any questions that pop up afterwards. Our email is in the chat, genealogy@acpl.info. And so for our next question, so for cases that might be one test away from solving, this person's asking which test, they say male or female tests are needed. So autosomal tests, Y-DNA tests, is there a certain one that is usually needed or a certain type of match?
We often will just stick with autosomal DNA just because every time you have to do a different DNA test with different DNA, that makes it cost more because every DNA test costs us money to do that analysis. So GEDmatch and Family Tree DNA, those are the 2 companies we're talking about. They accept autosomal DNA tests. So if you did a test at Ancestry or 23andMe or MyHeritage, you can download your DNA from there and bring it to GEDmatch and Family Tree DNA and opt into law enforcement matching. And it doesn't matter with what we're talking about, autosomal DNA, male, female, as many people as we can get into those databases, that was what is going to help us solve these cases.
Thank you. And our last one, are the recent terms of service changes at Family Tree DNA related to FIGG? If you might be able to speak from that.
I don't know when the last time they updated their terms and services. I will definitely have to check that because I know my course is starting again in October, and that's one of the things that I talk about is the terms and services, and I always have to go back and make sure I have a clip of like what the latest when it was last updated and see what the changes are. I mean, I think every site, I mean, even Ancestry updates their terms and services often. I don't know if there was something specific in their terms and services that was that maybe because of FIGG, but I think that that's just normal for any site to update them. And as far as I can tell, it doesn't change anything in relation to, you know, people uploading. It's just giving people more information about what what's allowed, what's not allowed, and I don't think it's limiting things in any way, or opening things up in any way that that wasn't previously allowed. Maybe it's just clarifying more.
Thank you. Thank you so much for your time and your presentation and for answering our questions this evening. So, and thank you to everybody for attending. Again, if you'd like a copy of the chat or if you have any other questions that pop up, our email is genealogy@acpl.info. So thank you again, Jennifer.
Thanks for having me.
Good night.
Good night.